Good evening, everyone. I'm Brock Reeve, the Executive Director of the Harvard Stem Cell Institute, and we're happy to host this evening's discussion as part of Hub Week. So Hub Week is brought to you by Harvard, MIT, the Boston Globe, and Mass General Hospital, and is a first-of-its-kind civic collaboration. And it really aims to engage the greater Boston community in the intersection of art, science, and technology. And in the course of the week, over 130 organizations have come together to celebrate the future, both in and of Boston, by hosting open studios, film festivals, we're not doing that tonight, a couple of short videos though, workshops, challenges, hands-on demos, conversations, and various competitions. This year, Harvard programs will explore pathways for increased civic engagement, advances within life sciences, and reimagined uses for the arts. We're thrilled to contribute to this university and citywide collaboration with the HSCI series Horizons in Regenerative Medicine and this evening's program entitled Honey, I Shrunk the Patient. To give you a quick background, the Stem Cell Institute at Harvard is a collaborative of more than 1,000 scientists and physicians dedicated to harnessing the power of stem cells to advance scientific and medical breakthroughs. Using it with our unique model of part research institute, part venture capitalists, we're advancing the study of diseases such as diabetes, ALS, you'll hear about that tonight, Parkinson's, and many others, where we believe stem cell biology is essential to bringing new treatments to patients. Today, we have an exceptional panel that represents many important players in the Boston biotech ecosystem who have found new ways to study and test human cells outside the body to help accelerate the pace of therapeutic drug discovery and lower the risk thereof. From disease in a dish to organs on a chip, these scientists are finding ways to shrink not only the human response, but also the time to patient for new drugs as well. To lead that discussion, we're happy to have STAT's Gideon Gill with us. Gideon is managing editor for Enterprise and Partnerships for STAT, a new online publication reporting on medicine, health, and life sciences. He oversees STAT's investigative journalism and special projects. He previously was the Boston Globe's Health and Science editor for a decade and had a hand in 3 Pulitzer Prizes. He was a Knight Science Fellow at MIT and has degrees from Harvard and Columbia in the process. Gideon.
Hi, thanks very much for that. Over the next 90 minutes or so, we're going to have a great panel here, and each of the panelists will first give us a brief talk about their own work, and then we're going to have a discussion. And I'll ask some questions to begin with to get it going, but then we really hope and encourage you in the audience to give us your own questions. And you all should have gotten index cards that you can write your questions on, and they will be collected. How we're gonna, you can pass them to the end of the row and we'll collect them. And in addition, you can also submit questions via Twitter because we are so technologically advanced here. And please use the hashtag HSCI Hub, HSCIHUB. One bit of unfortunate news is that one of our panelists, unfortunately, it was at the last minute fell ill and is unable to join us. That's Dr. Geraldine Hamilton, who is President and Chief Scientific Officer of Emulate, which is involved in commercializing organ-on-a-chip technology. But so we're sorry she won't be here, but believe me, we have a stellar panel, and we're going to start off hearing from Dr. Stephen Hyman, who is the director of the Stanley Center for Psychiatric Research at the Broad Institute of MIT and Harvard. He's also Harvard University Distinguished Service Professor of Stem Cell and Regenerative Biology. He's a principal faculty member of the Harvard Stem Cell Institute, and he was previously provost of Harvard and director of the National Institute of Mental Health. Steve?
So I'm going to start by stage setting. Why is it that we have to shrink the patient, and why do we need ways of understanding human biology? So you heard from Gideon that I direct a center focused on psychiatric disorders, and we're studying autism, schizophrenia, and bipolar disorder. And what we want to do is to exploit the amazing advances, which I'll touch on in a minute, in the human genetics of these disorders to come up with better therapies. So why do we need better therapies? Well, for one thing, all of the drugs that we currently use to treat schizophrenia and bipolar disorder are more than I mean, there's some new formulations and people have moved a few chemical groups around to make drugs safer, but the fundamental mechanisms are more than half a century old. So the mainstay of treating bipolar disorder is still lithium, which was first discovered to have therapeutic properties in 1949. It wasn't approved in the US until 1970 because nobody could figure out how to make a profit from an element. Somebody finally, obviously cracked that nut. The first antipsychotic drug, thorazine, was being experimented on in 1951 in France by a surgeon who was using it as a pre-anesthetic, that is to stabilize patients before surgery. And he said, wow, this stuff is so sedating. And he called up his buddies, Jean Delay and Denneker at a mental hospital and said, why don't you try this on your patients? And lo and behold, the sedation was in some ways a red herring. Over several weeks, what they saw is that the hallucinations and delusions melted away. The problem is that every single one of the drugs we have, even drugs that were approved last year, have the same fundamental mechanism of this drug. They block dopamine receptors. And the antidepressants is really the same story. I mean, iproniazid has a nice story in that isoniazid, very similar name, very similar compound, was still used to treat tuberculosis. And there was a clinical trial of a slightly different chemical form, replace the S with a P and move a few chemical groups. And it really didn't impress the bacteria, the mycobacteria. But in these sanatoria, I don't know how many of you have read Magic Mountain, there were a lot of people who were there chronically, they were ill, they were depressed, and they saw that this brightened people's moods, and lo and behold, the first antidepressant was born. But again, the mechanisms really haven't moved. So that's really terrible. So why has it been so slow? Well, first of all, the human brain is inviolate, it sits behind our skull. Cancer is a hard problem, as we know. Maybe you'll have heard some talks about cancer during Hub Week, but a surgeon does an excisional biopsy and hands a scientist the cells. For brain disorders, we can't do this, right? This is actually an Inca skull. They engaged in trephination to allow evil spirits to exit. The FDA has not approved this, And we don't have access to brain tissue, but even if we could do this rather impractical and unethical thing, it wouldn't really help because the symptoms, hallucinations or delusions of grandiosity, don't come from a single cell type or spot in the brain. They have to do with widely distributed circuits. So it's a hard problem. And then it turns out, and this is in some ways the example that will lead to a lot of the discussion tonight, we can put single genes into rodents and do all kinds of things, but in the end, it's the humans that have the disease. We cannot model it in rodents. I mean, it's going to sound silly, but what would a schizophrenic mouse look like, and how would we know? And are they hearing squeaks? And we laugh, but the scientific literature was full of these nonsensical creatures. Sadly, I still get papers to review that have them. We've got to do better. These are terrible illnesses, and we don't have good treatments. So here's an interesting clue that has just been sitting around hiding in plain sight forever, which is that we know that these illnesses run in families. And heritability is a funny measure. It's really the percentage of the difference between people that is explained by genes versus environment. And these numbers are an overestimate because they're taken from comparing identical twins with fraternal twins, and identical twins probably have a more similar environment than fraternal twins. But even with this inflation, the heritability of autism, even if it's half of this, that 40% of the cause of autism, or 50%, is due to genes. Same for schizophrenia, for bipolar disorder. I put up depression. It's interesting. It's much less heritable. It's much more heterogeneous. The environment, lived experience, has much more to say about depression. And so, being in recognition of how difficult it's going to be to solve these diseases, we started with nothing is low-hanging fruit, but these are easier than this. And schizophrenia is such a salient phenotype. People in their mid-teens, late teens start cognitive decline, and then they have hallucinations and delusions. We can pick these people out even though there's no blood test or easy diagnosis, whereas again, depression, we all know about it. We all know people, we may have had it ourselves. Very, very heterogeneous disease. So we've studied these things. Now, if you took genetics in high school or college, you learned about Mendel's peas. And Mendel's peas are really an example for very, very little in human disease biology. Remember, in Mendel, and part of the reason that he got these results is apparently he was clever enough, or maybe a little dishonest enough, to ignore anything that didn't reach integer values. But one gene would decide whether a pea plant was short or tall, peas were wrinkled or smooth, yellow or green, flowers white or pink. But in fact, these common human illnesses are not due to one gene. There's no schizophrenia gene. It turns out they're due across populations to many hundreds of genes. And why is that? Well, diseases like schizophrenia and autism decrease, people sometimes say evolution has stopped. Hardly. People with schizophrenia or autism, early onset diseases, have decreased likelihood of having children. They don't, in most societies, they don't partner, marry, have kids. And so any genes of large effect that were contributing to these disorders wouldn't be transmitted. They wash out of the population. So instead, we have a situation where there are hundreds and hundreds of genes, each one little tweaks, increasing the risk of these illnesses by 5% or 8%. And what happens is somebody gets an unlucky combination, and then there's some environmental effect that we don't really understand yet, because the environment's harder to investigate than genes, not because we're uninterested. And you get these illnesses. And the way we've gone about finding these needles, these tiny little effects in a haystack, is by becoming a brute force machine. And that makes me the brute in a brute force machine. But to give you some example, using gene chips, and I don't want to lapse into technical jargon, we've now genotyped about 60,000 people with schizophrenia and twice as many healthy comparison controls. And by the end of this year, actually, we'll have sequenced all of the protein coding genes in 20,000 people and so forth. And we're a little bit behind in autism. But we have these large global collaborations, China, Japan, Sub-Saharan Africa, South America. And in the end, we'll probably do genetic analysis on 250,000 people with each of these diseases and controls. And this is called a Manhattan plot, which, because it's supposed to have skyscrapers, and this is anything above this line is likely to be a true result. And this just shows you that with nearly 40,000 cases of schizophrenia, just genotyping on a gene chip, and again, I know it's not explaining all that much, we already find 108 places in the genome that are responsible for schizophrenia. So the goal is, a tragedy, as a scientist, it's great, it's a paper in Nature, and, even might get covered in Stat if we're really lucky. But it would be a tragedy if we ended up with a list of genes. What we want are disease mechanisms that people can then use to come up with therapies. But how do we do that? Right? So we know how to transfer single genes into a mouse, genes of large effect, but we don't know how to transfer scores or hundreds of genes into a mouse. And anyway, mice are so different from humans, right? Here's a mouse brain Obviously, the sizes are incomparably different. The scale is wrong, but here's a human brain. There's very little prefrontal cortex. In schizophrenia, the prefrontal cortex is the most damaged part of the brain, and that's the part of the brain we use for planning and a lot of higher cognitive function. So even if we found a way to transfer, to humanize a mouse at 100 loci with human genes, doesn't sound like a great project, still their brains are so different. Humans we're primates, right? We are diurnal, we are social, and we are visual. Rodents are mostly nocturnal. They are olfactory. In fact, many laboratory mouse strains turn out to be blind. They're olfactory, and they do socialize, but if we socialized in the manner of mice, we would be arrested because, they sniff each other's rear ends and things like that. So what are we going to do? And this is really to show you the necessity of what Kevin and presumably Adam are going to talk about, which is we need human genomes and we need human cells. And so we've started by taking little skin punch biopsies from lots and lots of patients and from lots and lots of healthy people. And the strategy is to take, first of all, and we collaborate, so Kevin and his team have done whole genome sequencing of many, many lines of these cells, because when there are all these little risk genes, we all have some level of risk genes, right? And it's true for schizophrenia, and it's true for autism, and it's true for type 2 diabetes, and it's true for inflammatory bowel disease. What matters is a pileup of these genes. And by the way, in other combinations, they're probably neutral, and in some cases, we actually think beneficial. In fact, we have some evidence that some genes associated with autism are also, in a positive sense, potentially associated with educational attainment, right? So, so we sequence these, and then we can take people who are healthy, and we can imagine adding genes or versions of genes that might confer risk of schizophrenia, of autism, of diabetes, and then we have to be smart enough to figure out what questions to ask the cell, right? What can we assay the cell? How will the cell be different? And then we can take patient cells, and we can make cultures, and we can begin to subtract genes and see, if we can find differences. But it turns out, and won't surprise anybody here, this is pretty reductive. We can learn a lot, but especially if we think about autism, we want to understand how the brain develops. Autism, probably, even though it's not diagnosed often until the age of 2, things are already happening at the time of birth. So can we begin to see brains developing abnormally? So our colleague Paola Arlada, other colleague Lee Rubin, have begun to make what are called brain organoids. So under the right conditions, if you raise any cell with any genotype that you're interested in under the right conditions, it starts to make a little brain. And Paola has now raised these little, they're not critters, but for as long as 14 months, and we're just beginning to ask the question, if we make a brain organoid with a background that gave a child autism and intellectual disability and seizures, anything like that, versus some comparison control that's healthy, are all of the right cells present? Are they expressing the same genes? It's early days, but I think what we've decided is that mouse, we're going to use genetic mouse models and other models to probe basic mechanisms. But in the end, especially for brain disease, but not only for brain disease, we need human genomes, we need human cells. And we're at the beginnings, I don't want to overpromise, but we're at the beginnings of, I think, a very exciting time that's going to change the way we are able to investigate these disorders. And with that, let me turn it over. Gideon, you want to? Yeah, you want to introduce Kevin and say how distinguished he is? Yeah. Okay. Yeah.
Question.
Yeah.
Which is, so what is, if you could give an example of what it is we've worked with mice and rodents, all this, and we test drugs on them, and there are lots of drugs that, I think it's fairly well known. There are a lot of drugs that work brilliantly in animals. We've cured just about every disease known to mankind.
It's never been
in mice, right?
right. It's never been a better time to be a mouse with Alzheimer's disease.
So, and which really, and then they get into the clinical trials and they don't work.
So I think this is what we're grappling with. And look, this is early. I don't want to overpromise. We have to be very careful. But we also have to learn from long failure. And mouse genomes are really different. We're separated by 90 million years in evolution. Again, we're diurnal and social and visual, and that means even the Darwinian environment acting on us has been shaping our genomes very differently. We have obviously very different cognition and behavior, and the idea that we can replicate one of these diseases in a mouse was, well, we were hopeful, but it didn't work. And I think it's really important, again, not to overpromise, but to say, brain disease especially is so hard because we don't have access. So can we begin to model it, literally, the title of this symposium? Can we shrink the patient and learn something meaningful that we can then take into humans with these illnesses?
Yeah, and so is the idea that eventually we would, instead of testing potential drugs on mice, we would look at try it on a brain organoid?
Well, yes, exactly. Now, I have to tell you, I just held a workshop at the National Academies 2 weeks ago, and the FDA was there, and the European equivalent, the EMA, was there, and they basically said, we don't care if you have an animal disease model. What we need to know from animals is safety. And even safety, it's really interesting. There, of course, there are a lot of drugs that look okay in animals, and we go into clinical trials. So people are also trying to make livers and kidneys to supplement animal testing for safety as well. Because again, we're just, we're very different from mice.
Yeah.
Right.
Thanks very much, Steve. Yeah.
Good.
So next, we're very fortunate to have Dr. Kevin Eggen here. He's professor of stem cell and regenerative biology at Harvard, also a principal faculty member of the Stem Cell Institute, and a director of stem cell biology at the Stanley Center at the Broad with Steve. In 2006, he was awarded a MacArthur Foundation Genius Grant, so that tells you something about him. Kevin.
So as Steve said, one of the challenges that we're currently grappling with is actually attempting to model these polygenic disorders like autism and schizophrenia. And it is a daunting challenge. And I want to provide, I hope with the support of some slides, some evidence that we've been gradually, over the last couple of years, building up to this moment. So Steve laid out this almost miraculous vision where we'd be able to make many different cell types of the brain from anyone that we were interested in studying who carried any combination of genetic changes that might predispose them to disease. So how is this possible? Well, it's possible through a series of breakthrough stem cell technologies, which are just celebrating their 10th anniversary today. And although there is a lot to learn about these technologies, they've already begun to show their utility in real ways. So how do we do this? It relies on taking easy to obtain cells like a skin punch biopsy or a drop of your blood, and to take these run-of-the-mill cells, which don't normally have the ability to become much of anything else, and expose them to a series of genes which are normally expressed only very, very early in development of the embryo. And remarkably, and I think what has to be one of the seminal discoveries thus far of this decade in the life sciences, these proteins, these genes can reprogram those easily obtained cells into what have been shown now to largely be the equivalent of embryonic stem cells, these more controversially derived cells from discarded IVF embryos. And those cells are special because they have two salient properties. First, they can grow indefinitely in the lab. So literally, if we were patient enough, we could fill this entire room with those cells, whereas normally your skin cells or blood cells would only divide a few times before they would sort of poop out. The other amazing property of those cells is that they can be coaxed to, in principle, become all the different cell types of your body or your brain. And we're still very much learning how to do that. I think it's important to say we're not experts in it, and we're learning how to take those cells and even make tissues like these brain organoids that Steve described. But it's exciting days following on from this. Also taking a lead from this type of approach, people are also beginning to realize more about the genes that are expressed in these differentiated cell types within the brain that dictate their identities. And people are more and more actually experimenting with taking cells directly from your body and turning them into brain cells for studies in the same way through a method called transdifferentiation. These are both really promising approaches for making cell types from the brain that we'd never been able to access before except through trepanation, as Steve said, for studies in the lab. Now, I want to say one of the first diseases that we chose to study in this way, almost to kind of prove a point, as I'll say in a moment, was a terrible neurodevelopmental disorder, which is kind of one of Mendel's P's in humans called Lesch-Nyhan syndrome. So this is a rare disorder which is caused by a mutation in a single gene, which encodes an enzyme which is important for recycling metabolites that are used in cells for making DNA, like guanine and guanine 5' monophosphate. And it's still not entirely clear what the mechanisms are that cause people to have very early onset changes in their brain, self-mutilating behavior, eventually motor neuron disease, and a variety of other terrible side effects that will eventually take their lives very early. It's also been proposed that the buildup of these products leads to an accumulation of uric acid, which leads to painful crystallization in different parts of their body that may participate in this. So this is caused by the removal of just this one gene due to mutation in these boys, a mutation that's on the X chromosome. Well, it probably should have concerned the entire field of mouse modeling right from its sort of nascence, because the very first gene that people ever mutated in a mouse using laboratory techniques was a mutation in this gene with the hopes that that mouse would get sick with this disease. Except for it didn't. And it lived a completely normal life, and it was totally normal. And to this day, no one has been able to detect a single change in this animal that would at all in any way, shape, or resemble any of the changes that exist in that person. And the thought is that evolution just simply took a different turn with respect to metabolism of these components within cells, and so the mice just have a way to get around this. Okay? So we wondered whether or not cells, even simple cells, would get sick in a way that might be more like these people. And so one of the first patients that we actually took cells from and produced these stem cell lines from was a young man who actually had this mutation in this gene called HPRT. And we compared their neurons to neurons that we made from a healthy boy. And you can see in this picture, there's quite a strong difference in the appearance and the number of these neurons that we can make in culture. And you can see here that this isn't just true for this one set of patients, but we see this strong change in a number of individuals that have this mutation. Now, we know this isn't just some accident, because we can take these boys' neurons and using a virus, deliver this enzyme back to that boy's neurons and restore their normal functionality. And you can see when we use an empty virus by itself without that enzyme, there's no beneficial effect. So we can be really sure that we're really modeling some aspect of this boy's disorder. This is kind of the simplest approach, and we've been gradually ratcheting up the difficulty and trying to look at more complicated disorders. One of the next things we've turned our attention to is a terrible disease called amyotrophic lateral sclerosis. You may know it as Lou Gehrig's disease. This is a terrible, fatal motor neuron disease, which is characterized by a gradual spreading paralysis that takes away the ability of people to breathe and eventually their lives. And the thing that's unusual about this disease is that there's one very specific neural circuit which dies within this disorder, a circuit that connects the brain to the musculature through a small number of neurons. And even though we've known about this disease for more than 100 years, we still have little idea exactly why this suite of neurons is degenerating in these patients. The best information that we have, and actually an inspiration for the type of approach that's been being taken in psychiatric disease and autism has again been success in genetics, giving us some sense of the genes that may be underlying this. And here the picture is a little bit different. So instead of many different mutations having small effects and adding up to autism or schizophrenia or bipolar, here it seems that ALS patients can have any one of many distinct mutations that each individually probably drive their disease. So in other words, this ALS patient's likely to have this mutation causing their disease, and this other patient's likely to have this mutation causing their disease, and those rare large effect variants are probably making people sick. And this is because unlike those disorders which really take away people's social ability to reproduce, these degenerative effects don't usually strike until after you've had kids, so they're not evolutionarily chased out of the population. Nevertheless, I could tell you all about these genes, but none of them would come into a coherent story about why that group of neurons is getting sick, and it's something that we've been trying to understand. Now, one interesting approach that people are trying to take with stem cell biology has been to ask whether or not aspects of clinical phenotypes that you see in people could be modeled in some way within the laboratory, and that's something that we've been trying to do. Neurologists in Australia, and first in the lab of Steve Vusick, has been using new techniques in electrophysiology in patients to actually try to make recordings of neurons in the brains of ALS patients. And what they found in simplistic terms with this method is that the brains of ALS patients have more activity than those of normal control individuals. And so first with Clifford Wolf, and now increasingly with Adam Cohen, as he'll tell you, we've been trying to measure motor neurons and other neuronal types that we make from patients with ALS to see whether or not there might be any difference from controls and whether or not we can understand them. And I'll show you just one example of this type of experiment that we've performed. This is now looking at the activity of neurons which come from a large number of control individuals and individuals that get sick because of different mutations which are causing their disease. And what I hope you can see is that there's this intriguing shared pattern of too much activity within these patients with ALS that we've been trying to understand. Experiments like this have led to the appreciation of new potential mechanisms which could be driving these changes and the investigation of new potential therapeutics using advanced measurement technologies, which Adam will say more about.
Thank you very much for that. I wanted to just ask you, so you talked about the various, that ALS, for example, there are many different genes that each can be driving this. So does this present a challenge when you're trying to model human disease when you have multiple genes involved? And Steve's also talked about with schizophrenia, many genes that each contribute a small amount. How do we possibly create enough models to understand that?
Well, simply speaking, there probably aren't enough cages here at Harvard to create genetic models for all the different forms of ALS and really be able to study them. I'll give you a great sort of extension of this metaphor that was used that's very specific to ALS. So it was really now more than 20 years ago that Bob Brown and colleagues first here at Harvard discovered that in a small number of families, mutations in this gene, SOD1, which I show you here, were causing ALS in a heritable fashion. Bob and others took mutant forms of that gene and forced mice to express many thousands of extra copies of that gene, and eventually they expressed enough that it made them sick. And people have studied that as a model for ALS for many, many years. There are two challenges to that. One is that mutation is only found in about 2% of people who get sick with ALS. And so if we find something going wrong in that model, we have no idea whether or not it is really relevant to the other 98% of people who are getting sick. So far, many things have ameliorated disease in that model, and that outcome has been 100% predictive for failure in the clinic. And in fact, the only drug that we have for ALS was discovered before the mouse model was made. So one could argue that we did better without it. We don't know, again, in this case, whether or not that's because there's differences in mouse and human physiology, which are making this problem a la Leishman syndrome, or whether or not we're just not forcing ourselves to do clinical trials on only people who have SOD1 mutations. Maybe that would work, but no one's bothered to have the resolve to actually try to do that. They just try to extend things right to the whole group of patients. And those people could be getting sick for an entirely different reason that we don't understand.
Thank you. Finally, I'd like to introduce Dr. Adam Cohen. Adam is a professor of chemistry and chemical biology and physics at Harvard. He's also a member of the Harvard Stem Cell Institute's principal faculty. He's a Howard Hughes Medical Institute investigator. Tech Review named him one of the top 35 U.S. technological innovators under the age of 35. And Popular Science named him one of their brilliant 10 top young scientists. So it must be true because it was in the media. And so Adam.
Thanks, Gideon, and thanks to all of you for coming here. That Tech Review thing, it's expired now. I'm over 35, so it doesn't apply anymore. So I'm going to tell you today about the next challenge that we run into when we have these neuronal models of disease, which is the challenge of trying to visualize the function, to actually understand what these neurons are doing in the dish. So I want you to take a minute and think about what's going on in your head right now. Each of us has about 86 billion neurons in our brains. And these neurons are connected to each other, each one to about 10,000 other neurons. And these neurons signal to each other with little electrical impulses about 1/10 of a volt tall and about 1/1000th of a second long. Okay? And so every thought and feeling and emotion, experience, memory that you have is encoded in these electrical impulses zipping through your brain. That's all there is. Right? And so to study these neural diseases, we'd really like to be able to see these impulses. The challenge is that we can't see electricity in a cell any more than you can see the electricity in this wire. You can't tell if there's a voltage here or not. And so it's very difficult to find drugs that can restore a healthy firing pattern if we can't measure the underlying activity. Comparing to the analogy that Steve made earlier to cancer, in cancer our goal is to either kill the cells or to get them to stop dividing, which is a relatively straightforward thing to measure. Here we're trying to coax one pattern of subtle and undetectable activity into a different pattern, and that's a much more challenging task. Traditionally, people have measured electrical activity in cells by taking a little glass capillary and poking it into the cell, and then connecting it to an electrical amplifier, and you can record these spikes. But this only records the activity at one cell at a time, or if you're really, really good at this, you can maybe get a few cells. This is a little bit like watching a movie by looking at one pixel on your computer screen. And you can see flickering when there's activity going on, but you don't get the full picture. To really understand how dynamics occur in the brain, we'd like to see the whole movie to see the patterns of activity among many cells. And so for decades, scientists have been trying to find ways to convert the electrical activity in cells into light, into something that we can directly see. And so I'll tell you a little story about our efforts in that direction. And the story starts here. This is a picture of the Dead Sea between Israel and Jordan. There's a single-celled microorganism that lives here in the water. This organism produces a protein which it uses to convert sunlight into electrical energy, which it uses to power its metabolism. Okay? So this protein sits in the membranes of these cells. It absorbs sunlight. And you can think of it like the world's smallest photovoltaic device. Light comes in and a voltage comes out. So a few years ago, I said to myself, I wonder if we can run these proteins in reverse. Instead of having light come in and a voltage come out, can we use the electrical activity in a cell to produce some kind of an optical signal, some kind of light that we can see? And so the idea is we'll put this protein in the cell, we'll excite it with a laser, and then when there's a change in the voltage, we'll see the light come out. And you see this works in PowerPoint, and so that's extremely encouraging. Okay. So after now I'm going to skip several years of hard work and just tell you that we found a way to do this. We discovered that these proteins are a little bit fluorescent, and their fluorescence reports on the electrical activity of the cells. There's another creature which lives in a freshwater pond in the south of England. which produces a different protein, which it uses to sense the sunlight so that it knows when to swim up and when to swim down. This other protein, it's called a Channelrhodopsin, it sits in the membrane, and when it's activated by blue light, it opens a little pore in the membrane, which can pass ions through, which then can deliver an electrical impulse to the cell. You can take the gene for this Channelrhodopsin and put it into another cell, like a neuron, and then you can trigger that cell to fire by shining blue light on it. Because ions flow in, it generates an electrical impulse, which makes the cell go. And so I thought it would be fun to combine this blue light activated Channelrhodopsin with the red light activated voltage reporter so that we could stimulate the cells with one color of light and record from them with a different color of light. So this way, it's like having a bidirectional optical interface between our instrument and the cells. And the neat thing about doing this with light is that you can control where you shine the light. So you can stimulate a subcellular region or one cell within a circuit. And you can image the whole response. This is an example. This is a rat neuron in a dish where we're optically targeting the blue laser just to the center of the cell. And then we're mapping the electrical impulse that's evoked as it propagates through the cell. And in this movie, we're mapping the propagation of the wavefront at 100,000 frames a second, so at extremely high speed. One of the challenges here is that these impulses in our brain, they're really short. Our brains do a lot of stuff in a very little time. And so we have to image very fast in order to see these impulses. And so here's an example of the kind of data that we're taking. Okay. And so you can actually see now in detail how these impulses propagate through the cell. Here's another example. This is on a different instrument. This is from a very wide field of view. So here it's about 1 millimeter by 3 millimeters. There's about 50 or so rat neurons here in this field of view. And I'll show you a movie where at a certain point we'll turn on a blue light over the whole field of view, and we'll just stimulate all of them in parallel. This movie is at 500 frames a second, slowed down about 10-fold so that we can see what's going on. And so when the light goes on, now you can actually see the cells light up, and you can see them start to fire, and you can see the activity in the dish. And you can go in there with the laser beam and stimulate just one of these cells, and then watch that one go, and then watch the signals go to the others, and then see an inhibitory cell, and sort of watch the whole circuit function. So you can really use this to tease out how these circuits work. I should say that interpreting this data is still a major and largely unresolved challenge. So coming out of these movies, we get data at about 1 gigabyte per second coming off the camera. And we have to segment that to find the individual cells. They're all overlapped. I think of these cultures a little bit like spaghetti and meatballs, where the meatballs are the cell bodies, and then the spaghetti is all of the axons and dendrites all interwoven with each other. And we're trying to sort of computationally disentangle the spaghetti. And that's a hard problem. And then when we see the firing patterns of the cells, we then have to interpret this. And connecting the activity of the cells, even in this sort of simplified preparation, to the underlying proteins and ions which are moving around in these cells is still a largely unsolved challenge. So that's work that still needs to be done. Now, these are rodent neurons. And as both Kevin and Steve explained, we're really interested in looking at human neurons. And initially, I asked my students if anyone would just give a brain biopsy so that we could measure these cells in the dish. And they were all strangely reluctant. And then I met Kevin, and my eyes got open to the wonders of this stem cell biology. And so we can take, as Kevin said, either skin or blood from patients and make stem cells, and then convert those stem cells into different types of neurons, which we can study in the dish. So Kevin showed the same photo of a ALS patient. And in collaboration with Kevin, so in Kevin's lab, they took stem cells from such patients and made motor neurons, or the cell type afflicted in the disease. And then in my lab, we've optically studied these neurons. And so here, I'll just show you a little movie of some human motor neurons in the dish. And as we stimulate them, we can see them firing away. And we can use this to try to understand how neurons derived from a patient might be different from neurons derived from a healthy person, and then how we can try to we can use this then to test drugs to try to find drugs that restore healthy patterns of activity. And Gideon, you asked Kevin this question about, well, but there's so many different people, each person has a unique constellation of genetic variations. How do you account for that? And I think the ultimate dream here is that you can do this procedure on anybody. And so for each patient, you could imagine if the disease is severe enough and there's enough motivation, making a stem cell-based model and then studying that very person's neurons in the dish. And the ultimate goal, which is still quite a ways off, is to test drugs on that patient's cells and to try to use that to predict which drugs might be effective in the person. I'm sure many people here have been through the experience of a doctor saying, well, let's try this drug and see if it works. And if that one doesn't work, let's try another one, and sort of going through this odyssey. And the hope is that we might be able to shorten that procedure. Because one thing we have to remember is that, so in a disease like ALS, where Kevin showed this pie chart of all of the different underlying mutations, just because we have a name for something doesn't actually make it a thing. And so ALS is a word which describes a set of symptoms, degeneration of the motor neurons, but it doesn't say anything about what's causing it. And in people, there are many, many different things which can ultimately lead to similar symptoms. And so by making these models from individual patients, we hope that we can get the right biology for that particular person. So here's a photo of some beautiful neurons in the dish. And I think that there's a lot of opportunities now to combine these technologies from stem cell biology and physics and chemistry together to try to develop better cures. Thank you.
Let me just ask you a question about, so this stuff's really cool. I mean, it really is kind of mind-blowing. And yet, what we're looking at here, the electrical activity visualized is just one characteristic of a cell.
Absolutely.
And so my question is, can this tool by itself tell you enough about the cell of a person with the disease versus a healthy person to really guide, to understand what's going on and to guide treatment?
Yeah, that's a great question. So this electrophysiology, as we call it, these patterns of activity, are very information rich. There's a lot of things you can glean by reading those spikes and looking at their patterns, but it's not the whole story. There's synaptic transmission, there's metabolism is very important, and there's a lot of other biochemistry that you would need to study in order to really do a complete job on disease modeling. This electrical activity turns out to integrate the dynamics of many different proteins. And so it does tell you about a set of things going on, but it's certainly not the whole picture. Another thing that we're missing here is these neurons, as we've plated them out in the dish, are wired up more or less randomly. And in some diseases, the disease acts in a large part through affecting the excitability of the individual cells. So for instance, in some forms of epilepsy, in some forms of pain, there's a reasonable chance that you can capture it in a model like this. But in many other diseases, autism, schizophrenia, depression, the disease is probably something about how the whole circuit is functioning in the real brain. And we've totally lost that here. And so the next real challenge, which Geraldine would have talked about, is to try to grow things which have more brain-like wiring, and then to apply similar technologies there to see what's going on. But of course, that's still quite a hard challenge.
Right. Thank you very much.
Okay.
So I'll start off by asking a few questions, but please, if you have questions, write them out on index cards, pass them to the end of the row, and we'll get to as many as we can. So I think starting off, I just wanted to ask, and each of you maybe can take a crack at this, but we've all trashed animals as models, so we can all agree that they've steered us wrong, particularly with the brain diseases, in psychiatric diseases. But one thing that the animal testing does, and actually, Adam, you alluded to this about we've lost sort of all the complexity of, and so can we, the animal at least has lots of complexity and it's all still intact. So can we really hope to get to the point where we have enough of the complexity there that these models actually work closely enough to how they would actually work in a real brain, with all the myriad cell types and connections of a brain that we can actually get real answers?
So let me start actually by defending those cute little mice for a minute. The mistake is thinking that you can turn a mouse into a good model for a human disease and then test drugs in that whole mouse that will predict efficacy in disease. That's a really bad question. On the other hand, how do we even have an idea of where to begin, whether to look at ion channels or action potentials or circuits? So animals remain critical for basic science experiments relevant to disease mechanisms. So I just wrote an editorial that won't make me any friends. It's going to come out, but it's entitled "Back to Basics," which is in the recent zeal of the science community to come up with cures. Many people have been racing to call this sad-looking mouse a model of depression when it's really a mouse that's been stressed by human investigators, or putting multiple copies of SOD1 into a mouse, or it's interesting, in Alzheimer's, we can give mice plaques and tangles, and then we find drugs that can do away with plaques and tangles. But the mice don't have Alzheimer's disease, and we don't know whether the human cognition gets preserved. So it's important to be able to produce plaques and tangles and learn what we can in the mice. But we should stop kidding ourselves that is then a model of human disease, right? We need to make different kinds of models that are more human. And you've asked a really hard question. How do we begin to capture the right level of complexity? And I can say that's why we're so interested in making brain organoids and little brainlets on a chip. But I think we're a long way off. But I think what we shouldn't do is fool ourselves again because we have a cool new technology. We have to be really tough-minded about what is going to make a difference for people who have these terrible illnesses.
Yeah, I mean, I think one of the problems is that scientists have tended to really ignore clear hints that they should not have moved forward in mouse modeling in particular areas. The Alzheimer's story is a great example. I talked about Lesch-Nyhan syndrome. Actually, even SOD1 is a great example. Yeah. So those mutations in ALS patients were found very early in the days of gene sequencing. Actually, some of the first positionally cloned genes that were responsible for human disease. And in fact, I think at the time, the mouse gene had not yet been cloned and sequenced. And so it was a very bad day for a graduate student somewhere when they cloned and sequenced the mouse gene, and they found that the mouse gene's normal sequence carries a sequence that would give you or me ALS. Okay?
Right.
And that didn't stop people from saying, oh, well, we should just give them more and see what happens.
Yeah.
Okay? And I think the problem is that then that kind of worked a little bit.
Yeah.
And the mice got sick with something that kind of looked a little bit like ALS. But then we kind of really have to say, well, does that mouse kind of pass the sniff test? And I think in retrospect, we really have to wonder whether or not that's true. And
that wasn't the best metaphor you ever
heard. But I think that's happened again and again and again. And but nevertheless, that's what people had at the time, and so it seemed the best opportunity.
Yeah.
we'll have to be careful about how we proceed. We have a
Right.
different problem. We have the right genes. We may or may not have the right cells. I
Right.
think we're still really
That's right.
facing
Yep.
that in the brain. And then, of course, we have to face the fact that it's this very reductional system thus far. These organoid technologies are new. They're exciting, but they're also in their infancy. And so we'll have to progress with great care and honesty that this is new. We definitely all agree. We need a new approach. We have to say this could be it.
Yeah, for every tool, you can use it well and you can use it poorly. And I think the same is true for these disease models, that the intact circuitry in the mouse brain is useful for exploring how things function in circuits. And the stem cell models are useful for exploring human genes. And I suspect in many cases we'll end up using both. And if you can sort of triangulate, and if things look good in both of these models, then that might be a more encouraging motivation to go forward.
We have a question here submitted via Facebook from Mary Holm. And she wants to know how and what do you base your decisions on deciding which diseases you want to model? Anybody want to take a crack at that?
Yeah.
At first I wanted to work on Parkinson's disease, but no one would give me any money to do that, except for HSCI, actually. And so we didn't get very far because the Parkinson's disease research community was really huddled around a small number of existing investigators, and they thought this was too crazy. And they were probably right. But the ALS community really felt that they were beginning to discover a variety of genes were causing the disease, and they were worried about the failures in the SOD1 model. They wanted to support a new approach. That, plus the fact that spinal motor neurons, which were one of the neuronal types affected in the disease, was one of the first cell types that we really knew how to produce from stem cells, is affected. And so those two things kind of came together in an opportunity to move into that space. And so we chose pragmatically, and then we became fascinated with the problem. And also one can't help but really come to care about people who are suffering from ALS, who essentially today have no hope. One becomes compelled to action over time in this area. So that's just one. That's examples of.
That's a good example. I mean, for the psychiatric disorders, once it became clear that they were highly polygenic and that the rodent genetic background was so different, we actually had no choice. It wouldn't have been the place to start. I think single gene disorders like ALS of the kind you describe, SOD1, C9orf, are the place to begin. But the study of these polygenic disorders has followed rapidly.
Yeah, the problem is that when you don't know the constellation of variants that are making you sick, you're counting on the experimental system to be robust enough and reproducible enough that without that knowledge and without the ability to pull the levers and switches on that, for it to be able to give you a robust and reproducible answer. So, if we think that this particular aspect of neurobiology is changed by those many little disturbances in our genome, then two things have to be true. One, we really have to master these techniques, and two, we have to be able to assemble enough people, enough normal individuals, and enough people who have these disorders so that we can see what normal variation looks like in these outputs, and then really be sure that when we see a change in a large number of patients, that we're really on the right track. Otherwise, we could fool ourselves.
Yep, I agree.
This idea, for a long time, people talked about bioprinting tissues and organs, and that received a lot of publicity. It still hasn't worked. And now we're talking about organ-on-a-chip technology and organoids, which is all really exciting. But all of you have talked, quite transparently, which I applaud you for, about that we're still very early on in this technology. But what gives you the confidence that it can work?
Well, In 2 days, my wife and I are going to get an ultrasound for her upcoming baby. And so we know it can work, and it's just a matter of getting the right conditions. And it happens all the time. And so I think if you can set the right boundary conditions and create the right permissive environment and the right nutrients, then we know that these cells have the capability within them to make a full-fledged brain. And so we have lots of examples.
Yeah. The Harvard compliance people are having chest pain because it sounds like you want to make babies in vitro.
Aldous Huxley would be pleased with that answer.
But it's true. It's really interesting. The idea of printing cells sounded great. It turns out that if you spin them and let them pattern themselves, they make they can make an organoid. If they're flat and you're interfering with just the geometry and the forces on the cells, they become something else. It's early days, but people are trying lots of different experiments, and I think, as Adam said, we want to find the right conditions. Yeah.
The other thing that gives me hope is another benefit of these advances in sequencing technology and the falling costs of sequencing. They're not just applied to characterizing our genetic makeup anymore. They can actually be used to catalog all of the messages that are sent out from our genes to make the proteins that, that compose our cells. And this means that one can, at a population level, from a tissue or a piece of the brain, or actually individual cells from the brain, actually read out the complete instruction list that cells are generating at any given time. And this gives an incredible quantitative picture of what cells are trying to be at any given moment, how disease genotypes change that, that instruction list. And I think that this is helping us. As far as cell production and making cells from stem cells, it's giving us quantitative metrics that we can try to live up to. And for some cell types, we do very well. Others, we're not there yet.
Yeah.
And this is going to be helpful. And I think the thing that will be most helpful for is that, the fact of the matter is, until recently, and I think Sangeeta Bhatia has been a real leader in this area, one of our colleagues from MIT, people still are trying to make liver and other, what you would call simpler organs from stem cells. And I think that's one thing that's let them down is that Sangeeta and others have shown that if you just take liver out of the body and you try to culture it in a dish, it changes very substantially. And so if you can't take liver out of the body and culture it and keep it liver, how can you culture something and hope to turn it into something like liver in a culture dish?
Yeah.
And so that's still a big challenge, and one that really needs the attention of engineers, biologists, geneticists. I think she's really realized that in her space and has been using those types of approaches to solve the problem.
A question for you, Kevin, which is that, so the neurons that you make from patient skin, basically iPS cells, how can you be sure that they realistically represent the patient's native disease neurons?
Well, I can tell you that what we make now are an approximation of young neurons, and really I would say the age, if you will, of those neurons, and our certainty about exactly what type of neuron they are really differs based on the recipes that we're trying to use. So for instance, the developmental biology around how you build the spinal cord is very well trodden territory thanks to extremely hard work by people like Tom Jessel and his colleagues at Columbia University. It's also one of the simplest parts of the nervous system from the point of view of a wiring diagram. So really following that recipe that the developing embryo follows has been very successful for making the right type of neuron there. We still think that those neurons represent sort of the thing that you would have at about the time that you were born from a best case perspective, or maybe early in your postnatal development, an already approved anti-epileptic drug based on the mechanisms that were at work in those cultured neurons. which could reduce that overall activity. It's something that we've explored with Adam as well. And the first thing that GSK wanted to know, since they're the owners of that drug and the marketers of that drug, is that if we just go back in healthy people and look at that electrophysiological marker that I showed you with the sort of flying saucer over that person's head, would that drug have the desired effect in that assay? And fortunately for us, it did. In that very simple reductional cell model, there was good alignment. But that was because we thought carefully about what the cell model was looking at. We thought carefully about what the in-patient assay was measuring, and those two things were able to line up. There are still many things that could have flummoxed that result, and we still don't know how that will work in ALS patients and whether or not their biology will be more complicated than we've been able to model in culture. But at least that's one example of from patient to cells to a drug to a success back in people, and we'll see whether or not that holds up in patients.
Well, I think we are out of time. Thank you all very much for coming. I want Brock to have the last word.
The only last word I have is to add my thank you, and please join me in thanking the panel and Gideon for kindly moderating it, and encourage you to stay engage in other Hub Week activities at hubweek.org, engage in following the work that we're doing at the Stem Cell Institute at HSCI online, and feel free, if people have a few minutes extra to stay and chat with people, feel free to do that. And thank you very much for joining us.